Thanks so much for inviting me. Yeah, I'm Terra Stein. I just started here 1 month ago in the Department of MedChem, and my group is interested in all things designing molecules. So I'm interested in small molecule inhibitor design as well as protein engineering, and really interested in how understanding dynamics and using AI can help us do these designs. And so I have both a computational group as well as a wet lab, and I'm really interested in really incorporating these two capabilities together and using computational modeling to develop hypotheses for testing in a lab, and also using experimental data in order to train our computational models. So I'm going to try and explain a little bit about how I would do this by talking about the research that I've done during my PhD and postdoc. So I did my PhD at UC San Diego with Mike Burkhart and Andy McCammon. And the Burkhart group is really interested in natural product biosynthesis pathways. And these are interesting pathways because they are creating molecules that have pharmaceutical relevance. They're also interested in re-engineering these pathways to create biofuels and just in general having bacteria synthesize molecules for us. So I'm going to tell you a little bit about this very canonical biosynthetic pathway that you may have heard of, fatty acid biosynthesis. So just as a very general overview, we have these we have these two carbon, oh, it was on the off, not on. No, it just doesn't go on the screen. Okay. Anyway, so we have these two carbon ketogenic units that are being added to a growing acyl chain, and these are interacting with subsequent enzymes to reduce this ketogenic group until you reach a fully saturated acyl chain. And one thing that you should pay attention to is that while these intermediates are being shuttled from the next enzyme in the pathway, they're always covalently attached to this acyl carrier protein. So this acyl carrier protein covalently has these ligands, and it's shuttling them to the next enzyme in the pathway. And not only is it interacting with these central enzymes, it's also interacting with many other enzymes that sequester acyl chains of specific lengths within the pathway. So acyl carrier protein has a really important role in these pathways, and there's a lot of analogous natural product pathways, for example, polyketide synthesis and non-ribosomal peptides. And so they have this really interesting way of delivering substrates. So initially, the substrate is sequestered inside the pocket of the protein, and then when it interacts with an enzyme, it has to deliver the substrate through this mechanism that is termed chain flipping. So we don't think that this is going to be a stochastic process where it's just going to randomly interact with enzymes, deliver substrate until it finds the appropriate substrate enzyme pair, because there's just too many possible combinations. So we think that there's some sort of regulation mechanism, and the hypothesis that we went into this with is that, let me see if my mouse, oh, I can use my mouse maybe, is that the identity of this substrate is going to change the conformation of the pocket, and this in turn will change the conformation of the external binding interface, therefore causing a difference in protein-protein interactions, and this is likely going to determine whether or not chain flipping should occur. So this is the hypothesis we went in with. And so something that's already known is that as you increase the length of the chain, the thioester is susceptible to hydrolysis. So the reasoning, the thought behind this is that the pocket can only accommodate a certain chain length. Once it gets longer, it's going to hairpin out, and then it's more solvent accessible, so it's going to be hydrolyzed. So in the group, what they've done is develop these chemoenzymatic methods to replace this diester with something more stable, like an amide bond, and they're using this for crystallography. But I'm really interested in dynamics, and so I was looking at these different products in the NMR tube, and what I found was that they're actually really, really different. So this was completely unexpected, because only the identity of that bond is changed. So the sulfur is being replaced with an amide or an oxygen, and all of the residues on the protein backbone, so each of these is about a backbone amide, and these residues are being perturbed. So they're experiencing a different chemical environment just with this one atom difference. And we can plot this and look at where this exists on the structure, and we can see that actually the part of the protein that's experiencing the most difference in chemical environment is actually the binding interface. So I also did some MD simulations to investigate a little bit more into like the 3D structure and the dynamics of why are they so different. One thing that I found is that there's this, the amide provides an additional hydrogen, right? So there's this additional hydrogen bond with this threonine 39, and that may help to explain why there's such a huge chemical shift perturbation at this location. And then taking the MD structures and just preliminarily docking them to a partner enzyme, you can already see that there's a different docking orientation. So this kind of goes in line with what our hypotheses are, but suffice it to say, I really wanted to study this in the most natural system possible. So I wanted to figure out how can we use this diester bond. And so I developed this method to do NMR by incorporating tiny amounts of this enzyme, so small enough that it doesn't actually perturb the structure of the ACP, but it's able to reattach fatty acids as they get hydrolyzed off. So we were able to use this in order to acquire NMR spectra of all of the biological chain lengths, and we noticed this interesting pattern as well. So most of the residues ended up clustering around the same general location, but there were certain residues that showed a really strong correlation pattern with the increase in chain. So about 10 of these residues, and these are occurring all across the protein, and we're really thinking that these residues are likely important for communicating the identity of the substrate, specifically the length of its chain. Again, I did some MD simulations to look at how this is occurring, and a lot of these residues we were able to actually pinpoint where exactly these residues are correlated with chain length. So this E57, for example, is actually uncurling as the length of the chain is increasing. So we think these are important for communicating chain length. We were also able to actually visualize this phenomena that was thought of as the reason why the thioester is so susceptible and why it's increasing with chain length, because it's really in the hydrophobic pocket, and then as it gets longer, it gets more solvent exposed, and we can actually get a visual on this hairpin formation, which was just really cool for us to see. And yeah, quantifying the pocket volume as well. Okay, so back to the drawing board. Okay, so we're pretty sure that this is true, right? When we have a different substrate in here, the pocket is different, the surface is different, but how do we know if this is actually affecting the protein-protein interactions? So the way that we tested that was we took this one enzyme, which is highly specific. In order to create lipoic acid, it only will perform catalysis with C8 chain length ACP. So we did this other NMR experiment where we took both C8 ACP and C12 ACP, which is not meant to perform catalysis with, and we saw, okay, wow, there's like a completely different protein-protein interaction profile. And so this was really exciting. And, you know, I brought this to group meeting and I was like, you guys, like, I'm really showing it. Like, it's really communicating the identity of the substrate. It's got different protein-protein interactions. And they said, yeah, but how can you really show that it's not doing the chain flipping? And I was like, okay, this is going to be really difficult to show, because how do we visualize whether chain flipping is occurring? And studies that they had done before involved attaching these fluorescent molecules of these solvatic chromatic dyes, and I didn't want to work with something really bulky like that because I already know how much like single atom changes are going to affect the structure. So I designed this really cool, I thought was really cool, NMR experiment where I specifically, everything is completely unlabeled, it's natural abundance isotopes, and then we specifically isotopically labeled one methyl group. So there's only one isotope label here. And so we were able to do this C13 NMR, and we see, okay, there's this one peak corresponding to the one labeled atom, or the one labeled chemical group. And then we added the LitB, and this was like really exciting. We saw that if you have the C8 ACP, you can see evolution of this entirely new peak, about 50% of the intensity. And when you have C12, there's no change at all. So that was really exciting to see, like, that we can visualize chain flipping, and that we can see that it really is only happening with the correct substrate. So then we went to look a little bit further at why this was happening. You know, we have these computational models. We know which residues are important. Can we try and alter the specificity of this enzyme using just the protein-protein interface? So I did, again, just docking the MD conformations using just really basic ClusPro, and saw that this E57 residue, which we saw was behaving differently in the NMR, as well as the MD simulations, we could see that the helix was curling outward. And you can see when this helix is curled outward here, it's not forming this salt bridge with this LitB residue, this arginine 145. So we said, okay, what if we just mutate this arginine 145? So we're just changing one residue on the surface. This is not close to the active site of the protein at all. And we were able to see that now the chain length specificity, it's not like as perfect of an experiment as the last exciting one, but like we can see that we're able to modulate chain length specificity just by engineering or creating mutation at the protein-protein interface. So now we have this C12 actually flipping into the substrate pocket of the LitB, which we didn't have before. So now I am really excited, and I want to start actually doing some engineering. And so we look at this system. So like I said, you know, fatty acid biosynthesis is not the only pathway that's doing this kind of mechanism. We also have analogous pathways. So there's this other pathway involved in polyurine biosynthesis, where instead of an acyl carrier protein, it's a carrier protein that's using amino acids to create non-ribosomal peptides. So this carrier protein actually attaches, gets attached with a proline. So we wanted to say, okay, well, what if we mutate this PLTF enzyme at the interface so that it interacts with our acyl carrier protein? And then we can create this product that's never been seen in nature. So how are we going to do that? And so the way that we started looking at this is using Rosetta. So I was doing the Rosetta part of this. Another student in the group, Josh Corpus, was doing all the experimental validation for this project. So we were working back and forth like this. So we took Rosetta, and we knew already that Rosetta is this really powerful protein modeling software that can do protein engineering. But we also knew that there were some flaws where the predictions are not necessarily always corresponding to what we see in the experiments. So we decided what we would do is we would generate mutations with Rosetta, we would test them experimentally, and then we would refine the score function and see if we could get some better models. So already though, with just using Rosetta and using this information that we had about the structure and communication, we were able to do iterative rounds of mutagenesis. So we found that this one mutant really significantly increased the activity. So the wild type had like already a slightly like very small amount of activity already, which I think was what made this possible to even do. but we already are getting like 40% activity. And what we did was we took the best mutants here, we put them through a second round to get double mutants, did this again to get triple mutants. And what I wanted to say is, okay, can we improve upon this? So can we modify the Rosetta score? So the way that the Rosetta score works is there are these terms that describe the interactions between atoms. So we have electrostatic, hydrogen bonding, van der Waals, and a lot of other terms. And so I did this at the time very basic, low throughput way of modifying the score function. And so I think that that the way that I tend to approach things, and I think is pretty common, is that, okay, I'll see if I can do it by hand, like in a way that's not so efficient, and then let me see if I can have the computer do it in a way that's more efficient, I think. So this is the less efficient way, just to see if this would work. And so what I did was I visually inspected the complexes of the mutants, and then I looked at the breakdown of the scoring terms, and I said, okay, well, which of these score terms is ordering the interface score in a way that aligns best with the experiment, right? So because we know, oh, this is another graph of the experiment, we know that the order should be, the order of activity is this, so which of the score terms is ordering it in the same way? And at first it looked like electrostatics and hydrogen bonds. We did a little bit more analysis on the second round and the third round, and saw that electrostatics is really most important, which was not unexpected because we know that ACP is a very negatively charged protein. So we wanted to increase the weight of the electrostatic score, and we tried a few different weights, and the one that did the best was this this weight of 1.5. And what we saw was, okay, now we're getting less false positives, or yeah, so like before, what would happen is the program would say, okay, we expect that these mutants are gonna perform well, or they're gonna have a binding interaction, but we saw in the experiment that they're not actually performing this reaction. And so we didn't want to get this, right, because we don't want to make these mutants go through all the process of doing cytokine mutagenesis if they're not going to be good. So we found that we were able to generate a lot more like true positives, and we were able to generate the most activity a lot quicker. So nowadays, if I were to approach that, I would do something similar to what they did in this paper, where, you know, we're using machine learning in order to reweight all of the terms. Like that would be maybe a more efficient way. And so that's what I did during my postdoc, which I'll talk about later, was learning about all these different types of machine learning architectures and how I can use them to advance my research forward more efficiently. And I'm just going to show you this slide one last time, because so the final thing that I was really, really interested in doing before I defended was understanding this chain flipping process, right? So I was able to visualize it in NMR. That was really exciting. Now I wanted to simulate it. And so the issue with simulating such a large conformational change is the issue that we encounter with a lot of molecular dynamics simulations, is that we end up only being able to, well, we only have capacity with current hardware and software to simulate with conventional molecular dynamics the around the microsecond time scale, maybe millisecond, if you have something like a really good computer. But we have this issue where we're ending up spending a lot of time in these low energy conformations, and it's quite difficult for us to access these transitions that occur on a slower time scale, so milliseconds and seconds. And these are a lot of times the biological processes that we're actually interested in. So we presume that the chain flipping is happening at the second time scale. And so I looked at a lot of different ways of doing enhanced sampling. And what a lot of the methods do is they end up altering the underlying force field, right? So they're applying a bias. So you're forcing you're modifying the force field in order to direct your stimulation to the place of interest. But what ends up happening here is you end up losing a lot of kinetic information. So you can observe the process you're interested in, but you're not really certain about the underlying kinetics. And so in order to, so we found one method that actually doesn't modify the force field at all, that's able to simulate transition paths in such a way that we felt like it was a good method to move forward with. And this is called the weighted ensemble. And so the way that this works is you're dividing your progress coordinates. So you have a start state and an end state, and you want to sample in between them. And you divide your conformational space into bins. And you start your simulation, they're all here in this low energy space, but very rarely you'll end up crossing a bin. And the way that this method works is in order to basically direct your computational resources towards simulating this process of interest, is you split up the trajectories that end up, and you replicate the trajectories that end up crossing a bin. And then what also ends up likely happening is that they'll fall back down into this low energy well. But what you want to do in that case is you just merge them so that you're not spending all of your computational resources. So this is a way to focus your computational resources towards the pathway of interest through doing these splitting and merging and keeping track of the probability of bin crossing, but you're not modifying force field at all. So you're able to still be able to calculate things like rate constants once you get a converged simulation. So we decided to use this enhanced sampling technique, and then something happened, which was called the pandemic, and you might have heard of it. And so this happened, and I had already done all of this, work. I had finally figured out, because this weighted ensemble method is a bit complicated. The software is complicated. I'm, really trying to see okay, how can I apply this? How can I, do this? And then this happens, and I realized that I could apply everything I just learned, all the benchmarks I've just done. I can reapply it to this other conformational change that many more people would also likely be interested in, other than myself and a few others in the field. And so Romy and Morrow at UCSD had done this really incredible job of developing these all-atom models of the glycosylated spike from SARS-CoV-2. And what I wanted to do was, and so something that you might know is that there's this cyan receptor binding domain. And the way that the spike works in order to infect cells is it has to go from this closed to open conformation. So this is the transition that we wanted to simulate, and this is required in order for it to bind to ACE2 and then infect human cells. And I'll just say a little bit about glycosylation. One thing that we already knew is that it helps to shield these viruses from the immune system by mimicking the glycosylation patterns that we have in our own proteins. And what they found was that there's also this structural role where if they just did the end state, the up state, they were able to show that the glycans were actually playing a role in holding up this receptor binding domain. But I wanted to understand the transition from the receptor binding domain. So sometimes the video needs a little bit of a push. Okay. And so we ended up doing this really huge collaboration and this really ambitious project, because this is the largest system that has ever been simulated with this method. It was 500,000 atoms, and we were collaborating, with the best, with Romy Amaro, with Lillian Chung, who develops the weighted ensemble software, WESPA, and we were using a lot of computational resources in order to simulate this pathway, and we were able to generate pathways of spike opening using about 100 V100 GPUs for a few months straight. So this is a really intense endeavor, but we were able to get like the full atomistic mechanism of spike opening and really understand all the residues. And we were able to find that this other glycan, which wasn't even really on the map in the other studies, is actually important for, it has a functional role, right? So we know they have this immune system role and the structural role, but this pink glycan, N343, is actually going underneath the receptor binding domain and pushing up the receptor binding domain in a way that you might be able to see if the video was playing more. Okay, so, oh, there we go. Just a little flip up. Okay, so we did that. That was really exciting, and we did this other collaboration with Joachim Frank, who is able to do this method with cryo-EM that's really interesting, called manifold EM, where they're taking different conformations from the experimental data set, and they're able to basically predict a conformational landscape. And we were able to actually overlay our MD simulations. We put them on the cryo-EM map and overlay the simulations. And this was the first time I really saw, like, you're really showing that the experiment and the simulation are doing the same type of dynamics. We also collaborated with Jason McClellan. And so basically we had predicted, we were able to have the whole atomistic mechanism. So we predicted these residues that were important for spike opening, and in particular, and so all of them did decrease spike opening and therefore ACE2 binding. And the glycan in particular decreased it by more than 50%. So this glycan is really important. And this was really, just a joy to collaborate with all these people. We did this was like a part of a bigger collaboration to simulate the entire virus, all atom, and we won the Gordon Bell Prize. And then again the next year, an even more ambitious project, a billion atom simulation to simulate the delta spike inside of a respiratory aerosol. So yeah, so now there's so yeah, a lot of research has kind of gone towards viruses, and then I defended my PhD, and I wanted to learn, like I said, machine learning, and I went to Berlin with Frank Noe, and I have been looking at using deep mutational scanning data of this receptor binding domain to the ACE2 receptor. So we have these, we have like 100,000 data points from this deep mutational scan where we have these different sequences, and we're able to, we have sequence and then we have a binding affinity. So because we have so many data points of sequence to binding affinity, we're able to create a neural network in order to predict this, and we're able to extrapolate to other sequences and really have an idea of predicting how well sequences are going to be able to bind to ACE2. And we use this to create an evolutionary model, and we're simulating the evolution. So using this neural network ACE2 binding affinity as a fitness function to say, okay, it's going to evolve more if it's continuing to get better at binding ACE2, and we're able to predict actually variants of concern that are seen, these delta, alpha. We're able to predict these without training up. So that was pretty exciting, and that's it's actually 31 minutes, so I'm going to wrap up right now, and I'm just going to say that, yeah, I'm here, and this is kind of something like the type of research I'm going to be doing, computer-aided drug design, so a lot of stuff with SARS-CoV-2, protease, which I didn't talk about, and protein engineering. I'm interested in hydrogenase systems for bioenergy, biofuel, and I'll talk more in the future about AI and simulation, particularly more things with the weighted ensemble method. Here's my lab right now. I have Jason Lynn, who's right here, and then I have 2 rotation students, Tiger and Raphael, who are here. So just starting out, but all the work that I presented today was in collaboration with a lot of other folks, so I have to thank them as well. And thanks for listening. I have a few questions.
Can you get the question?
With the sampling method, how do you choose the bands at a equal space?
So it's actually the information that you get is really independent, right? So you get the statistics are independent of the bin spacing. So we start out with equal bin spacing, but there's certain regions of conformational space that it's really easy to cross over, and in certain areas that it's really hard. So you want more finer spacing in that region. Yeah. Yeah, and then there are ways to do the automatic bin placement as well that are being worked on.
Information space is information.
Yeah.
So how do you define the bin?
Yeah, that's one of the big challenges. You want to find a progress coordinate that's going to efficiently describe your transition of interest. So here we're using oftentimes a 2-dimensional progress coordinate. But yeah, there's also a lot of interest in using machine learning. But then you're also evaluating the progress coordinate at every time step. So you want something that's going to be efficient. We also used RMSD, so it kind of incorporates like multiple degrees of freedom as well. Yeah.
Let's think one more time.